Okay, so welcome to the second video for our discussion on genetics and inheritance. So far we've kind of did an introduction of genes, alleles, dominance, recessive, genotypes, phenotypes, and Punnett squares. And so what we're going to do now is we're going to take that introductory terminology and knowledge and use it to look at work that was completed by Gregor Mendel in the 1850s, and use that to look at his work and understand patterns of inheritance and how he kind of figured out these patterns of inheritance. And so Gregor Mendel was an Austrian monk, um, who worked with pea plants in the 1850s. And he did a lot of experiments on these pea plants to determine how um, traits are passed from parent to offspring. And he came up with some basic rules of inheritance that we've already mentioned in our discussion on mitosis and meiosis. The first of those rules is the law of segregation. And so that's that your gametes can only carry one allele of each gene. So for example, if your genotype is dominant B and recessive B, your alleles, so let's say these are two sperm cells, the sperm is only going to carry either a dominant B or a recessive B. It can't carry both because mitosis, remember, goes from 2N, which is diploid, to haploid cells. So that's that law of segregation. So Mendel's work actually provided the foundation for discovering and determining segregation of gametes into alleles, or alleles into gametes. There you go. The second law is independent assortment. And this basically says that if you have genes for different traits, they separate from each other independently during meiosis. Now, this applies in most cases, and we'll talk about when this doesn't apply later on. But basically, let's say you have your hair trait, like we've been talking about in the first video. So brown hair versus blonde hair, dominant versus recessive. And then let's say we're looking at height. So let's say tall versus short. So dominant versus recessive, tallish and short. Now, if you are a tall individual with brown hair, that doesn't, if you have your offspring isn't necessarily going to be tall with brown hair. You could also, so your offspring could be short with brown hair, or they could be tall with blonde hair. So basically, if you pass on the dominant allele for hair color, so brown hair, that doesn't mean that you have to pass on the allele for being tall. You could pass on the allele for being short because they assort into the gametes independently of one another. And that's because, if you remember, your homologous chromosomes line up on the metaphase plate, right? And so this one would be big B, little b, this one would be big T, little t. And they go, so they could line up this way or they could be flipped, right? That independent assortment that we talked about. And so that's why it was important to understand it when we were talking about meiosis, because not only is it important for genetic variation, but we're coming back to that idea now. And so we've already talked about this extensively, but we just kind of want to bring it back up because, again, we're going to focus on dominant and recessive alleles for a big portion of this second video. So the dominant allele is suppressing or masking the complementary allele, which is generally the recessive allele. And so if you have two dominant alleles, so if it's homozygous dominant like this, then it will be the homozygous dominant trait, which in our example is brown hair. And then even if you have the heterozygous form, so dominant with the recessive, because it masks that recessive trait, you still get brown hair. You only get the recessive trait when you express that allele in homozygous recessive form, so like this. That is when you get your blonde hair. So it is not expressed if it's paired with a dominant allele, again, because of that scenario there, where dominant will always overexpress or mask, not overexpress, but suppress or mask that recessive allele. However, just because it's labeled as dominant does not mean it's more common than the recessive. Dominant alleles can be rare in the population. It's just that whenever it shows up with a recessive allele, it suppresses that allele. Okay, so dominant doesn't mean more common. It means it's dominant in terms of the expression when paired with the recessive allele. So here we have that Punnett square cross. And so what Mendel did is say he took two pea plants, and these pea plants can have different color peas. They can have yellow peas or green peas. And what he did is he took a pure breeding yellow pea plant and a pure breeding green pea plant. So what pure breeding means is that whenever you breed that plant, you will get that same trait over multiple generations, right? So if we do a Punnett square for capital Y, for dominant, homozygous dominant, right, with two individuals, you always get uppercut, homozygous dominant. And then if you take this offspring and breed them together, you always get the homozygous dominant. So it's pure breeding for yellow colors. Same thing for the green pea plant. If you always use your lowercase, you're always getting homozygous recessive, so always green peas. So these are pure breeding plants for their respective trait. Now, when you take those pure breeding plants and you hybridize them and you mate them together, so yellow pea plants with green pea plants, that first generation, okay, so this parent generation is called the P generation for pure breeding. Okay, now when you breed those two together, it gives you an F1 generation. The F1 generation will always be heterozygous individuals, because you will always have a dominant allele given by the dominant trait and a recessive allele given by the recessive trait. So you always have a dominant and a recessive. So 100% of the offspring will be heterozygous, and 100% of the offspring will have the dominant trait, which is yellow. So it seems like the green pea trait is lost, but really it's just being masked. And so if you take two individuals from this F1 generation and you mate them together, so heterozygous individuals, and make an F2 generation, this Punnett square shows you what you end up getting. So what you get is a 3-to-1 ratio for the phenotype. And remember, the phenotype is what it looks like. So 75%, so 3 out of 4 will be yellow peas. 1 out of 4 will be green peas. So for every 4 pea plants, 3 should be yellow, 1 should be green. But now for the genotype, you have a 1-to-2-to-1 ratio. So 1 genotype is homozygous dominant, 2 genotypes are heterozygous, and then the last one is homozygous recessive. This is always the case. If you take a dominant recessive trait and you breed a pure breeding generation, the F1 generation will always be heterozygous. And then if you breed for that F2 generation, you will always get these ratios. A 3-to-1 phenotype and a 1:2:1 genotype. These ratios are super important to remember, and these ratios show up on a monohybrid cross. Okay, so with one trait. All right, and remember this is dominant recessive. So we can see the same thing with widow's peak hairline. So widow's peak is the dominant trait, straight hairline is a recessive trait. So if you have two individuals that are heterozygous and you breed them together, their offspring is going to have a 3:1 phenotype, so 3 with widow's peak, 1 with straight line, and a 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So a 3:1 phenotype and a 1:2:1 genotype. Okay, so only 25% of their offspring will have a straight hairline. You can do the same thing for attached earlobes versus unattached. So you can see right here, it kind of attaches to the side of the head. Here, that lobe's not attached to the side of the head. We also have things like polydactyly, so various different traits that can be expressed through those patterns of inheritance. Okay, so that example was with a monohybrid cross. So looking at one trait, for example, pea color. Now we're going to look at two traits. And so two trait crosses are known as a dihybrid cross. Okay. And this is where the law of independent assortment is really important, because it says that alleles of different genes are distributed to gametes independently, so irregardless of what allele you get for other genes. Okay. And this only applies if those genes are on different chromosomes. If they are on the same chromosome, then they always travel together. So this is assuming that those two genes are on different chromosomes. And so we're going to use the earlobe and widow's peak example to show this dihybrid cross. So free earlobes, so non-attached, is dominant. Attached earlobes are recessive. Widow's peak is dominant. Straight hairline is recessive. So if we have a pure breeding individuals, right? So homozygous dominant and homozygous recessive. That means that because they are homozygous dominant for both, that they would have the dominant trait, and then homozygous recessive for both, they would have the recessive trait. So widow's peak, free earlobes, straight hairline, attached earlobes. Now, if they were to mate, all of their offspring will have the dominant traits because they would all have the heterozygous genotype for each trait, right? And so then if you take these heterozygous individuals and they mate, the chance that their offspring has one of the four combinations can be shown in this dihybrid cross. So this is the P generation, this is the F1, and this is the F2, okay? And what's important here is that the alleles are segregating independently of each other, okay? And so that tells you what gametes you can have. So for example, if the individual has this genotype, it can give an uppercase E or a lowercase e. If it has this genotype for widow's peak, it can give an uppercase W or a lowercase w, okay? And so what you can do is you can do what's called the FOIL method. So let's say this is the genotype, where you do first the inner, so that's EW, so that one, then this one, so you have that one, then you do the inner to inner, so it's that one, and then outer to outer, so E lowercase, which is that one. And so those are the four gametes you can have, the four combinations. And so since the male is also heterozygous for both, you would have exactly the same gamete combination. And then all you do is you combine them. So you have dominant E, which is there, dominant W there. So let's look at, say, this one. So you have a lowercase e with this first column, uppercase E. So you have heterozygous. And you have dominant W, so it's homozygous dominant. And so when you go through and you do all of these combinations, this is the Punnett square that you end up with. And so there are 4 potential combinations you can get here. You can have an individual with free earlobes and widow's peak. You can have an individual with attached earlobes and widow's peak. You can have an individual with free earlobes and a straight hairline. And you can have an individual with attached earlobes and a straight hairline. So this is homozygous dominant, right? This is homozygous. Let me erase this, make it clear. Chicken scratch, sorry. This is homorecessive, and then these are going to be, depending on which one, is going to be various versions, right? So some are going to be homozygous recessive, whereas others will have some dominant forms. And what you can do is you can go in and you can count these up, right? And so basically, with the F2 generation on a dihybrid cross, the phenotypic ratio for those 4 combinations is going to be 9 to 3 to 3 to 1. So 9 individuals will be dominant. So that is the free widow. 1 individual will be recessive, which is the attached straight. And then you'll get 3 for the other ones. So attached widow and free straight. So it would be 9, 3, 3, 1. So out of 16 possible combinations, this is that phenotype. And we can go through and we can check this out. So dominant. So dominant E is attached. Dominant W is widow's peak. So there's 1. So dominant. So we see those. There's 2. We got a dominant E, dominant W. So there's another one. Dominant. So there's 4, 5. So this is dominant and recessive. So this is not. So this is a free earlobe but a straight hairline. So that's not one we want. So this is free earlobe. So dominant. So this is 6. This is straight hairline. So we don't want that one. This one is there's no hair on that one. I've never noticed that. But this is dominant. So that would be 7. Then we've got dominant is 8. Not 6 is 8. So recessive, dominant is 9. And then we have recessive. So there are 9 individuals that are free earlobes with widow's peak. Now we can go in and we can say, okay, now which ones are going to be attached with widow's peak? So if they're attached, then they're recessive E. So they have to be homozygous recessive. So this one is attached with widow's peak. This one is attached with widow's peak. And this one is attached with widow's peak. And then we can go in and say, okay, which ones are going to be free earlobes, but with a straight hairline? So this one we want dominant E's with recessive W's. So this is 1, free ears, straight hair, 2, free ears, straight hair, and 3, free ears, straight hair. And then the last is homozygous recessive for both, which is this one. So that's where our ratio comes from, the 9 to 3 to 3 to 1. This ratio always happens when you go from a dihybrid cross with pure breeding to an F2. You always get that 9 to 3 to 3 to 1 ratio. The genotypic ratio is a little more complex, so we don't worry. It's just a bigger ratio number. So we typically don't really learn that. But the phenotypic ratio is very helpful. So 9 to 3 to 3 to 1 in terms of this F2 generation. Okay, and so we have this animation we're going to look at to kind of help with this if you still have some questions.
During the 19th century, the Austrian monk Gregor Mendel performed specific crosses with pea plants and worked out much of what we know today about the laws of heredity. We will use his ideas to consider the inheritance of a single trait, freckles. The inheritance of freckles is determined by a dominant allele. Suppose a freckled female who was homozygous dominant, big F, mated with a homozygous recessive male with no freckles, little f, little f. How likely is it that their child will have freckles? A Punnett square helps determine the probable outcome of the genetic cross. Both parents are homozygous, so each can produce only one type of gamete as far as freckles are concerned. The female produces gametes with the dominant allele, big F, and the male produces gametes with the recessive allele, little f. Each square in the resulting table is filled in by combining the alleles from the sperm and eggs. In this example, all the children would be heterozygous for the trait big F, little f, and therefore would have freckles. Now let's consider why it's possible for parents who are both heterozygous for the freckle trait to have a child without freckles. The dominant F and recessive F alleles segregate during meiosis, so half the gametes bear the dominant F allele and half bear the recessive F allele. Filling the boxes, we see that the probable genotypes of the children would be in a genotypic ratio of 1 homozygous dominant, big F, to 2 heterozygous, big F, little f, to 1 homozygous recessive, little f. Phenotypically, the children would be 3 freckled to 1 not freckled. A child from these parents would have a 25% chance of not having freckles. Suppose 2 people who are heterozygous for widow's peak and freckles mate. Freckles and a widow's peak are both controlled by dominant alleles, so these heterozygous individuals show both traits. Would it be possible for this couple to have a child that had neither freckles nor a widow's peak? We can use a Punnett square to analyze a 2-trait cross, as long as the genes for the 2 traits are on different chromosomes. Keep in mind that during gamete formation, the alleles for a gene segregate, and that genes for different traits that are located on different chromosomes will segregate independently of one another. Therefore, 4 possible allele combinations are produced in the gametes: dominant F, dominant W, dominant F, recessive W, recessive F, dominant W, and recessive F, recessive W. Each type of gamete has an equal chance of joining with any other type at fertilization. The relative numbers of phenotypes possible for the next generation are 9 freckled with widow's peak, to 3 freckled with straight hairline, to 3 no freckles with widow's peak, to 1 no freckles with straight hairline. The expected phenotypic ratio is 9 to 3 to 3 to 1.
Okay, and so another way to kind of look at the dihybrid crosses is if you understand that independent assortment, and so if you understand independent assortment, you understand how it works, then you know that you could do a monohybrid cross for each trait because they're independently assorting from one another. So basically, you can say, okay, let's look at earlobes and let's see what the frequency is going to be, and then let's look at hairline and what those frequencies are going to be. And you know, based off of the monohybrid cross, that the F2 generation is going to be a 3-to-1 ratio for phenotypes for both, and then a 1-to-2-to-1 genotypic ratio. And so if we want to know which ones are going to have free earlobes and widow's peak, then we can use those monohybrid crosses and you just take the product of them. So you can say, okay, if 3/4 is going to have free earlobes and 3/4 are going to have widow's peak, that means 9/16 is going to have free earlobes and widow's peak. And you can do that for attached earlobes and straight hairline. So 1/4 times 1/4, because of 1 out of 4 gives you 1/16. And then you can look at whether or not they're free earlobes. So if they're free earlobes, they're 3/4. And if they're a straight hairline, they're 1/4. So that's 3/16. And then if they're attached earlobes versus widow's peak, then that's 1/4 times 3/4 for 3/16. So 9,331. You can see that there. That's a 9,331 ratio. Okay. All right. And so that's it for the second video, where we're talking about monohybrid crosses, dihybrid crosses, and how we can kind of get at the different probabilities for complete dominance. So when you have a dominant and a recessive trait. What we're going to talk about in the next video is variations from complete dominance, so things like incomplete dominance, codominance, etc. And then also some diseases or some issues that you can get when the alleles don't segregate properly in meiosis, and you have gametes that have more than one copy of each allele, so say 2 copies, and kind of what that can lead to in terms of effects for the organism. And so as always, let me know if you have any questions, if you have any concerns, and we'll see you in the next video.